in the past years is also very important, and you know that as well, is just pharmacokinetics. Yes? So you can do a lot with binding kinetics biochemically in cells, but finally it's also you have to do it in an animal model or in humans, so pharmacokinetics remains very important.
Okay. So I need to ask Michael one question. So, one chance. Since I see you are working on your mobile. Yeah, so that's good. And we are over time. We had the question before for structure-activity relationship. What is the right assay to work with to advance your molecule to the next level? You know, you can start obviously with the isolated enzyme, or going all the way up to the cells or even to tissues, which would be physiologically more relevant, but you know, more complicated and less throughput and complicated structure-activity relationship. And I think you want to comment on this, right?
Yeah. Do I need to use a microphone or? No. So had you asked me that question 5-7 years ago, I would have said a functional assay, because I was then focusing on ion channels, and binding to an ion channel is not necessarily something that's going to give you the right answer. I think if I speak with my screening hat on as a drug discoverer, I would like that my primary assay, I mean, if I can have as few assays as possible. So if my primary assay can be my primary SAR assay, that would be the best. And again, with my screening hat on, I again would go for something that determines the function of your target enzyme. So that would be number one. But failing that, obviously the most informative in my mind SAR driving assay, once you're past your actives generation stage, could be indeed a cellular occupancy assay. And of course, it's doable to do that in sets. So the values are hard and quantifiable, but it is not an affinity measure. It is not a K-off. It is a SETSA value. So I think before you completely swap, I would like for more companies like Sanofi or AstraZeneca, the companies that have the muscle to start screening your diversity libraries, or at least publishing this kind of boring benchmarking data of this is what these 20,000 compounds look like in an enzyme activity assay, this is what it looks like in SETSA, and this is what it looks like in SPR, or something like that. So that we know when to pick what assay, because I don't think the SETSA is the solution to everybody's problems, but to quite a lot.
Okay. That's a fair statement. Thank you for your contribution. Great. If there are no further comments, I would like to close the panel.
Like to comment.
Yeah, okay, great.
So I think in the perfect situation, we should have both at the same time, both in vitro, because it might give the better view of what's going on in the, like how the protein works, how the molecule binds, maybe how the whole pathway works. But then, of course, it's really important to validate it in the physiologically relevant model. So this is, I would say that this is the perfect situation to work in parallel.
Okay, thank you. So with this, I think we can close the day. Thank you very much, the panelists, to be here on stage, giving your insight. Thank you to the audience participating very willingly to this discussion. That was great and enriched our discussion, of course. I think we had a very nice day today. It started with how to choose diseases to work on. We heard about modalities, what is out there. We heard about new lead generation technologies. We have heard about residence time and other new screening technology, testing technologies. So that I think was really a nice day which span from the very early phase to the later discovery phases. The technologies which are out there touch upon quite some material there, quite some subjects. So this brings us to the end of the first day. I just want to make one announcement. I will leave to a nice restaurant at 7 o'clock in the lobby. If you want to join me, I'm happy to take you with me. But everybody has to pay his own bill to set expectations right. So if you have the evening free and would like to talk to people, please come join us in the lobby. I need to know roughly who's considering to come with me to probably make a reservation. So if you could give me a hand sign just to have a number. Yeah. So 10, 12 people. Okay. So let's reconvene at 7 o'clock, and then we will move to this restaurant, which is nearby, so we will walk and it will take us 10 minutes. Okay, thank you very much for the nice day we had.